Thank you, Luis, for the introduction. Hello, everyone. I am Takuma Terada from CIMIC Swift Zero. Today, I will present first. Later on, I will hand over to Mr. Yuki Shinohara for his presentation. Today, we are going to present current trend in oncology development. First, I will present about precision medicine, overview of ADC development. Then Mr. Shinohara will present example of novel ADCs and immunotherapy development. I will start the presentation for precision medicine. Personalized cancer therapy is treatment strategy centered on the ability to predict which patients are most likely to respond to specific cancer therapies. This approach is founded upon the idea that tumor biomarkers are associated with patient prognosis and tumor response to therapy. In addition, patient genetic factors can be associated with drug metabolism, drug response, and drug toxicity. Personalized tumor molecular profiles, tumor disease site, and other patient characteristics are then potentially used for determining optimum individualized therapy options. This slide shows novel cancer drug approved by FDA in 2020. Most of these drugs are targeted therapy, including antibody drug conjugate, cytotoxic agent, and cytotoxic agent is limited. Also, we can see the various indications in this slide. Now, precision medicine is one of the most important perspectives in the oncology field. This is an example of the targeted therapy. As you know, NCCN guidelines for non-small cell lung cancer recommends molecular testing for EGFR, ALK, ROS1, BRAF, MET, RET, and PD-L1 for advanced or metastatic non-small cell lung cancer patients. Then we have several treatment options depending on the molecular profiles, as we can see here. On the other hand, genomic testing is underutilization in the real world. Dr. German presented it at the ASCO 2019. Patients who performed EGFR single testing is 54%. All testing such as EGFR, ALK, ROS1, and BRAF is only 22%. All 7 tested is only 7%. Even when driver mutation detected in the genomic testing, only 45% received biomarker-driven treatment. Tissue insufficiency, cost reimbursement challenges, poor performance status, and turnaround time may act as barriers to the uptake of comprehensive molecular testing. This data is from MSK Impact. During 2014 and 2016, 10,945 tumor samples sequenced and 36.7% of patients harbored at least one actionable alterations. However, genomically matched clinical trials were only 11%. This gap reflects systemic shortcomings in the availability and geographical accessibility of relevant trials, as well as patient preferences. Additionally, many patients may have been too debilitated to qualify for the clinical trial, and conversely, others who may later receive from targeted therapy continue to benefit from conventional treatment protocols. This data was reported by Dr. Tsunami from National Cancer Center Hospital, Japan. 58.2% of the patients harbored actionable gene aberrations, but only 13.4% received molecular targeted drugs in accordance with gene aberrations. The gaps between the number of patients with actionable mutations and those received genomically matched therapy indicate the need to develop drugs targeting new genes covering not only druggable kinase genes, but also non-kinase genes, such as epigenomic and transcriptional regular genes, which are often mutated in a variety of tumors. These results also may suggest patient performance status was not enough to receive targeted therapy due to the turnaround time. FDA approved liquid biopsy NGS companion diagnostic test in 2020, such as Guardant 360 and Foundation 1. Dr. Nakamura reported liquid biopsy NGS testing improves turnaround time, as you can see in the figure left. According to the data from GI Screen and Godzilla study, the turnaround time shrinks from 35 days to 12 days. Also, Dr. Nakamura reported actionable alterations are similarly identified by tissue and plasma genotyping. Trials enrollment rate in patients with actionable alterations are improved in the plasma testing cohort. This slide shows comparison of tissue biopsy and liquid biopsy. Tissue biopsy cannot be done for everyone due to the limited of tissue sample, but liquid biopsy can be done. Tissue biopsy can confirm histology, but liquid biopsy cannot. Turnaround time for liquid biopsy is faster than tissue. Evaluation of resistance heterogeneity for liquid biopsy is broader than, but tissue biopsy panel size is larger than liquid. We should consider pros and cons for each biopsy. Then I will move on to the next topics. Antibody drug conjugates are empowered antibodies designed to harness the targeting ability of monoclonal antibodies by linking them to cell-killing agents. An ideal ADC has a highly selective monoclonal antibody for tumor-associated antigen that has restricted or no expression on normal cells. A potent cytotoxic agent designed to induce target cell death after being internalized in tumor cell and released. A linker that is stable in circulation but releases cytotoxic agent in target cells. Although ADCs were initially designed to leverage the selectivity of monoclonal antibodies, improving the therapeutic index, there have been many challenges which have emerged during their development, resulting in a very large number of trials ultimately being halted due to the excessive toxicity. This is a history of approval of ADCs. Mylotarg is first approved in 2000, but voluntary withdrawal in 2010 following negative trials. Then brentuximab vedotin was approved in 2011. TDM1 was approved in 2013. Now we have 10 ADCs in the market. More than 80 ADCs are in clinical development with nearly 600 clinical trials ongoing. Combining ADCs with immune checkpoint inhibitors, T-cell agonists, and other agents that affect immune response has the potential of reverse many of the evasive strategies that tumors use to circumvent immunosurveillance. Currently, approximately 36 trials with 20 individual ADCs in combination with immuno-oncology therapies are ongoing, most of which are checkpoint inhibitors. From the next slide, Shinohara-san will talk about the examples of ADCs.
I'm Yuki Shinohara, Director of International Clinical Development from Untitled Chapter. From here, I would like to introduce several examples of ADCs. This is an example of novel ADC CX-2009 Probody Therapeutics. CD166 is broadly expressed in normal epithelium and overexpressed in many types of primary and metastatic malignancies. Normal tissues expressing CD166 at high levels include lung, pancreas, intestine, prostate, and liver. Given its high expression on normal tissues, CD166 hasn't been considered as a target for therapeutic development due to potential safety concerns. Probody Therapeutics, a recombinant antibody drugs designed to be activated by proteases in the tumor microenvironment and preferentially bind to the tumor rather than to healthy tissue. CX-2009 is a Probody drug conjugate that consists of a humanized anti-CD166 monoclonal antibody conjugated to DM4, a microtubule inhibitor. Preliminary data suggest a potential association between CD166 tumor expression levels and clinical activity, as in this slide. Next, please. This is also novel ADC DS8201, which has improved by FDA in last year for breast cancer. Although the target HER2 is well established, the payload deruxtecan is topoisomerase 1 inhibitor. Unlike trastuzumab emtansine, trastuzumab deruxtecan has a released payload that easily crosses the cell membrane, which potentially allows for a potent cytotoxic effect on neighboring tumor cells regardless of target expression. In addition, the released payload has a short half-life, which is designed to minimize systemic exposure. DS8201 shows response in several cancer types, as you can see in the slides, for colorectal cancer, breast cancer, and lung cancer and gastric cancer. Following the ADC, let's move on to the immuno-oncology. As for the trend of immuno-oncology, the number of IO drugs in the development pipeline in 2020 has grown to about 4,700. It was 22% increase compared with 2019 and more than 200% increase with 2017. On the clinicaltrials.gov database, over 6,000 active clinical trials that are testing IO agents identified, which shows 14% growth compared with 2019. The IO field continues to grow, with more drugs and trials for almost all IO drug classes compared with previous year. In the development of T-cell targeted immunomodulators, researchers have continued to try finding the effective combination of modality for changing cold tumor into hot tumor. Next, please. It is considered that immunologically hot and cold tumors differ in neoantigen burden. Hot tumors just need checkpoint inhibitors or combinations of checkpoint inhibitors and/or checkpoint agonist antibodies to optimize T-cell function. However, cold tumors require T-cell to be first primed and then to traffic to the tumor tissue in addition to checkpoint modulation. Several strategies exist to induce priming of T-cells and/or enhance antigen expression, such as vaccine, oncolytic virus, chemotherapy, and radiation. Next, please. Immunomodulators with chemotherapy led some of breakthroughs, such as for TNBC, hepatocellular carcinoma, and etc. Whereas the number of the cell therapies agents has increased rapidly compared to T-cell targeted immunomodulation. Among the different cell therapy types, CAR T-cell class has the largest increase, whereas novel T-cell approaches such as CRISPR-engineered T-cells or gamma delta T-cells and other cells have increased. Next, please. CAR-Ts are classified as autologous or allogeneic. A majority of cellular immunotherapies in development are autologous. However, the preclinical and phase 1 development of allogeneic therapies have raised. The top 10 targets for blood tumor indications are shown. CD19 is still the most dominant target for cell therapies against blood indications. But the number of active agents targeting B-cell maturation antigen, BCMA, or CD22 have nearly doubled since last year. Next, please. While the largest indication categories for solid tumors are covered of cell therapies for which the tumor-associated antigen, TAA, has not been disclosed, and the greatest change in the top 10 targets is for Glypican-3 and prostate-specific membrane antigen. The number of cell therapies in preclinical and clinical development continues to expand. Meanwhile, most allogeneic agents are in preclinical and early phase clinical development, and it may be some time until these agents achieve widespread clinical validation. As a summary of this session, next please. Precision medicine is crucial for improving patients' treatment in oncology field, but genomic testing is under utilization in the real world. ADCs are one of the options to improving the therapeutic index. Several novel ADCs are under development in clinical. IO field continues to grow, strategy become more multifaceted. CIMIC Group has a strong network with KOLs in Japan and has handled various complex clinical trials from early to late phase with quality and speed. We also support companies who wish to enter Japanese market and navigate them through local regulatory processes. Lastly, we'd like to introduce our company. CIMIC Group started out as a CRO assisting in clinical development of pharmaceuticals. And now we offer greatly expanded services providing added value from preclinical to commercialization. As we call the pharmaceutical value creator, with this business model, we also offer comprehensive solutions to pharmaceuticals, medical device manufacturers, academia, bioventures, and medical institutions. Next, please. This shows CIMIC's proven track record. We are the largest CRO in Japan with over 1,200 CRAs and has been involved in nearly 80% of the new drug development in Japan. We also have a clinical operation team in 12 Asia-Pacific countries, such as Korea, Taiwan, China, Singapore, and are connected with more than 2,600 clinical sites. If you would like to learn more about the oncology development in Japan, please visit our website where we have on-demand oncology webinars, or feel free to contact us. We didn't have enough time for the Q&A session today. Thank you for the questions, and we will get back to you separately after this meeting. Liz, I will hand it back to you.